Okay, today we're going to talk about DNA structure and function. And before we start, we'll talk about sort of the people that discovered the structure of DNA. And that's these two gentlemen on the right, James Watson and Francis Crick. They discovered the structure. That's the model they built in 1953, and they won a Nobel Prize for medicine for that. A woman that might not be a household name for you is this woman down here, Rosalind Franklin. She actually was the one that was doing the X-ray diffraction that was actually shooting X-rays at DNA and getting the images, and it's alleged that they, Watson and Crick, stole her images or got them without her permission, and that enabled them to put together this model and become celebrities. Unfortunately, she exposed herself to so much radiation getting these images of DNA that she died of cancer shortly after they made this discovery, so she never had any way to sort of contest their model. Anyway, let's just kind of go over the organization of DNA. Right? We talk about, DNA is this molecule, and there are chromosomes, and there are genes, and how's all this stuff related, right? So remember, when we talk about a gene is sort of a recipe to make a polypeptide. There are also regulatory genes that we'll talk about, but when we talk about genes, we're talking about typically a sequence of DNA that contains the code or the recipe to make a protein. Those are what we call structural genes. Then we have a whole sequence of genes on one piece of long DNA, and that's called a chromosome. So a chromosome is a one strand containing many genes. Now remember, chromosomes only look like this when they're in prophase and metaphase during cell division. Usually they're unraveled and they exist as chromatin. So usually when you look at a cell in the nucleus, you're not going to see chromosomes in this structure. With the replicated sister chromatids, you're going to see them unraveled as chromatin. Okay, but then the genome, you may have heard of the Human Genome Project. The genome is just all of the chromosomes, all of the genes on all the chromosomes in every one of your cells, right? So for humans, we have about 3 billion nucleotides. We'll talk about what a nucleotide is, which make up about 25,000 genes. They say between 20 and 25,000 genes. Actually, not that many genes on 46 chromosomes. So first off, how did they get to know about DNA as this sort of genetic material, this sort of blueprint of life? Well, it started in the 20s with a guy named Griffith, and he did experiments with bacteria, pneumonia bacteria, and mice. And he knew there was a pathogenic strand. It was pathogenic because it was covered with this protective capsule, and when he injected that into a mouse, the mouse died. And we looked at the blood of the mouse, it had this bacteria in it, so we could tell it died from that. He also knew that there were harmless strains that didn't have the protective capsule, which appeared rough when you looked at them under the microscope. And we injected that into the mouse remained healthy. Its body was able to attack and destroy the bacteria. He also knew if he heated up some of these capsulated pneumonia and injected them into the mouse would be fine, right? Because we know that heat denatures proteins, and the enzymes in the bacteria would not work, and that we wouldn't have a viable bacteria. Okay. But then he did something interesting. He mixed some of these heat-killed bacteria with some of the harmless living, right? And when he injected those into his mouse, the mouse died. Now that's odd, right? You'd expect the heat-killed bacteria are dead, they're not going to do anything, and the harmless bacteria are harmless, so they shouldn't do anything. But then when he examined the cells of the mouse, the blood of the mouse, he found that they had viable, pathogenic, coated, capsulated form of the pneumonia. So he came up with this idea of transformation. Something has transformed this harmless bacteria into something pathogenic, right? And at the time in the 20s, most people thought it was probably protein, because proteins do most of the important things in our bodies. They're enzymes, they're hormones, they control things, they're receptors, and so forth. So probably going to be proteins, right? But then in the 40s, two scientists, Hershey and Chase, did an experiment. They said, well, let's make sure it's not something else. So they went down to the simplest organism they could find, and that's a virus. A virus is basically a piece of protein on the outside, right? So that's protein. And then in the middle, there's some DNA. So they said, let's actually see, we know viruses can take over a host cell by injecting some kind of instructions into those cells, and the cells stop what they're doing and they just start producing viruses. So this must be the material that tells cells what to do. If they're able to hijack the cells, that must be what cells use for their instructions, right? And we know now that that's DNA, but of course they didn't have transmission electron microscopes to tell that, so they actually had to do an experiment involving a blender and some isotopes. So they started out with some viruses, and they exposed some of them to an isotope of sulfur, sulfur-35. Remember, sulfur is part of that tertiary structure of amino acids. So that sulfur was going to bind to the proteins within those viruses. Okay? And they incubated those viruses and then let them grow and assimilate that isotope of sulfur. And then they did the same thing with a phosphorus-32, a radioactive isotope of phosphorus, again exposing viruses to that, letting the viruses grow, and the phosphorus is found in DNA, so that's going to get incorporated into the DNA of the viruses. So then now they have viruses with radioactive proteins, and they have viruses with radioactive DNA, and they basically put them into flasks with bacteria, let them kind of attack the bacteria, insert their genetic material into them, right? And then after a period of time, they took them, they centrifuged them basically with a blender, and they agitated them with a blender, then they centrifuged them in a centrifuge machine, and that basically separated out the cells from the liquid. And what they found was, with the isotope of sulfur, the radioactivity was in the liquid and not in the bacteria. Okay? Whereas when they analyzed the phosphorus, they found that the radioactive phosphorus was in the bacterial cells and not in the liquid. So that they realized that what had happened is that the DNA had got injected into the cells, and that was the genetic material, and not proteins. Okay. So now looking at the structure of DNA, again built on what Watson and Crick discovered. Well, we know that it's made up of three parts: a base, either A, T, G, or C, a sugar called deoxyribose, and a phosphate. And the basic unit of that is called a nucleotide. So here's an example of a guanine nitrogen base attached to a deoxyribose sugar and attached to a phosphate, and that would be a guanine nucleotide. So it's basically the monomers that make up nucleic acids in DNA are nucleotides. Okay. Then we also know that DNA forms a double helix. A helix is a backbone. So we have one backbone there, one backbone there. The backbones are sugar phosphates, and then the ladder, the rungs of the ladder, the middle of the ladder, are the nitrogen bases, and this is where the code is within the DNA. And we always pair A to T and G to C. We'll talk about why that is in a minute. This is sort of a two-dimensional view of DNA, but we know that DNA has a three-dimensional view. It's more of like a spiral staircase than a ladder. So this shows sort of the three-dimensional view, and then if we actually show the sort of space taken up by each of the atoms, a space-filling model, it actually looks more like that, and this is sort of what the model was that Watson and Crick put together for DNA. Okay, so base pairing in DNA, we always pair A with T, and we always pair G with C. An easy way to remember that is A and T is made with straight lines, G's and C's are curves. And why is that? Well, you can see here that for them to bond together, we have these hydrogen bonds which hold them together. So a hydrogen across from an oxygen, again, opposite charged atoms attract. Nitrogen is also very electronegative, so it's going to be attracted to the positive hydrogen, and they're going to form two hydrogen bonds there between the A and the T. When we look at guanine and cytosine, we can see they form actually three hydrogen bonds because they have three sets there. So A's and T's bond together because that's how they'll form hydrogen bonds, and guanines and cytosines will form those three hydrogen bonds in their arrangement. Okay. And then the last thing, why A and T and G and C? Well, it's got to do with, again, if this molecule is a ladder and we want every rung of the ladder to be the right size, you can see that we can't have a purine, which is double ring, with another purine. That would be too wide. We can't have pyrimidine with pyrimidine. We have to have a purine with a pyrimidine. So we always have A with T C, they can form their hydrogen bonds, and they'll all be the right size, and that is why DNA has that structure. So this is just an overview of DNA. In the next screencast, we'll go over how DNA replicates.